Welcome to Plantopia. I'm your host, David Goodori, and I'm a plant pathologist at Cornell University. Any listeners who have never met a real live plant pathologist can be forgiven. We are a very low visibility, but very high impact profession. We protect the world's food supply from disease-causing fungi, bacteria, viruses, and nematodes, all of which want to eat your lunch. This time on Plantopia.
You need one person per 1,000 acres to harvest wheat. But in the case of strawberries, you need about one person per acre. People are looking at the guest worker program as the solution. And I tell them this is a short-term solution for what is a long-term problem.
Robots have long since made the transition from science fiction to a reality of our everyday life. Few of us, however, are familiar with the quiet revolution that has taken place in agriculture, the forces that required that revolution, and where it is taking us.
The stars have kind of aligned. We've got this tremendous need right now, and then the technology is coming together at the right time. We're gonna save the industry, I believe, and keep berries and other fruits affordable and available for U.S. consumers.
In all the previous work of the past 20 years, working on grape powdery mildew resistance around the world, in 1 year we doubled the number of resistance genes that are known.
In this, the first of 2 episodes on robotics, we'll take an in-depth look at why there is such an emphasis on applied robotics in so many areas of agriculture. We're going to start with the seemingly simple act of picking a strawberry. And from there, we'll learn how robotics can be paired with high-resolution optics and artificial intelligence to drastically accelerate the discovery of disease resistance genes.
Hi, I'm Lance Cadle-Davidson. I'm a grapevine pathologist with the USDA Agricultural Research Service.
Hi, I'm Gary Wishnatzki, owner and head pixie of Wish Farms.
What is Wish Farm, and where are you located?
Yeah, Wish Farms, we're a year-round grower and marketer of all 4 major berries. We're based in Plant City, Florida, right on Interstate 4, right between Tampa and Orlando.
And what are the 4 major berries?
Currently, the 4 major berries are strawberries, blueberries, raspberries, and blackberries. But we're actually getting ready to add a 5th one.
Can you tell us, or is that a national secret?
It's a little bit of a secret right now, but I'll let the cat out of the bag. The 5th berry is actually going to be the pine berry. It's a white strawberry that has hints of pineapple flavor, and it has like a little pink blush to it when it's ripe. Dr. Vance Whitaker, the plant breeder with the University of Florida, just released his first white strawberry cultivar, also known as a pine berry, with that little pineapple taste, and it's going to be awesome. It's we had some little small pilots last year and got really great acceptance in the marketplace, and I think it's going to be a major thing for the growers in the state of Florida.
I've watched strawberry harvest, and I've scrupulously kept away from being conscripted into the process because it looks like hard work to me. How are strawberries harvested now?
It is hard work. Try picking a white strawberry and figuring out when it's ripe. So currently all berries are being harvested by people. It's the thing that a lot of people don't understand is strawberries don't ripen all at once. They ripen over the course of, in Florida here, it's a 5 months or more season. So every 3rd to 4th day, you have to go through the field and pick off the berries that are at the right stage of ripeness and leave the green ones. And it's, if you don't get them at the right time, you can lose the entire crop sometimes just because of a disease can take over if you let berries get overripe on the plant.
And who does this work?
The current folks that are doing the work are predominantly right now H-2A guest workers, and we're bringing them in on visas. We're having to transport them, house them, and pay them a higher than what the national minimum wage is. It's an adverse effect wage we have to pay. And that's just the minimum. Folks are making, many are making in excess of $20 an hour. Our California farms are people making between $30 and $40 an hour in during peak harvest. So the money's not bad for what you would most people consider unskilled labor. However, to strawberry growers, it is a skill. I mean, in order to be able to make a good living at it, you have to be fast, you have to be accurate, you have to be able to stay in a field all day and stooped over picking strawberries. It is really hard work, and there is a skill to it. It's not exactly unskilled labor.
But this isn't a case that fits the stereotype of someone stealing a job from a hardworking American. They don't want to do this work, and somebody is willing to do it.
No, and it's true. I mean, even back during the Great Recession, we had people that would come out looking for work, and they would do it for a couple hours. If they made it to the second day, they were really an anomaly. You didn't see very many people that could make it past one day that were not used to doing the work. So, yeah, no, the average American, we're definitely not stealing jobs. And the thing that's also, I think, of note is I've been around the world and, at other places that grow strawberries, in any country that is first world, they all have labor that's coming from somewhere else. I know in England right now, in Great Britain, there's a lot of problems with labor. They had a program, Pick for Britain, and that didn't work out so well. With Brexit, they were having issues with getting in people from Eastern Europe that were traditionally doing the picking. And in Australia, they have people they call them backpackers there instead of migrant labor, but they're people that just follow the crop around. But mostly it's people coming from somewhere else. It's not the domestic people in the countries that are producing the berries that are picking them.
So we're kind of getting at the subject of today's podcast the long way around. And you had mentioned that the workforce is first-generation immigrants. But there's something about the demographics and the social aspects of this workforce that goes beyond the problems that we face today. Those problems are manageable, but the future doesn't look so bright. So can you tell me a little bit about that? What's going on?
I started noticing back in the early 2000s, there were less and less people showing up at the farm applying for work. And throughout the early late 2000s, it became quite evident that something was going on, that, there was just less people coming to do the job. And when I realized what it was a demographic issue. You look back at the population in Mexico and the fertility rate there, that's where all the people were coming from to do farm work. But back in the 1960s, the fertility rate there was 6.7. The average woman was having 6.7 children, big families, and they were plenty of people looking for work, and they were coming across the border because there wasn't work in their home country. But then I looked at, by 1995, that fertility rate had dropped below 3. And now today, it's almost at 2. It's just barely sustainable for their population, so if you looked at back in the early 2000s, we were seeing this shrinking workforce, and we're continuing to see it today. And that's why we're so heavily dependent on a guest worker program. That guest worker program was never designed to be a only means of getting workers. It was a means of last resort when growers didn't have the labor and available. But today, it's our only way to get the crop picked. And here's the thing. So where we're at today, that fertility rate in Mexico 20 years before this, back in 2000, was still, above that sustainable level, but it's been continuing to drop. So if you kind of look at that trend of where it was and what our workforce was, you can see why the workforce was dropping. But the next 20 years is going to be even more critical in my mind, because people are looking at the guest worker program as the solution. And I tell them this is a short-term solution for what is a long-term problem, because in the next few years, Mexico will soon become a net importer of labor. People will not be leaving Mexico to look for work. There's going to be work there. Farms in Mexico are going to be short labor. The bidding is going to keep going up. Currently, our labor costs have been skyrocketing, and the cost of getting berries harvested has been going up. And I'd say in the last 3 or 4 years, California is a good case in point. I mean, it used to be maybe a $9 a box break even. It's already up to $12 a box break even. And the pricing has been going up, but it's not going to keep up. It hasn't been keeping up with the cost of the inputs there. So the thing that is, to me, is kind of a little bit scary for the American consumers is the affordability and availability of fresh fruits and vegetables. The way that the costs are rising, growers are either going to have to get a lot more money for their products, or they're going to go out of business. And, it's just going to become a supply and demand thing. The industry is going to shrink, and the availability to consumers will not be there in the future unless we solve this problem.
And the problem's even more complicated than just the demographics might indicate, because those people who are being added to the workforce aren't necessarily interested in picking strawberries. If they're going to college to receive advanced education, their career goals probably don't include picking strawberries.
That is true. Yeah, folks don't raise their kids to be strawberry pickers. The first generation people that are coming here to this country that are still doing the work in the fields, they all have a vision for their kids to go on to do much greater things and to get an education and to go into other types of jobs that aren't as taxing. So it's not. That's why I say first generation people are doing the work, because the next generation don't want to do it, and quite frankly, they shouldn't. That's why people come to this country for opportunities, and the immigrants are always the ones that are willing to do the hard jobs that nobody else wants to do. That's been the history of our country.
Which finally brings us around to the subject of robotic harvesting.
That's my favorite subject.
So, I've seen this harvester in action, and unfortunately, this is not a visual medium. So, could you describe it for us?
So, it's a pretty large vehicle. It's about 30,000 pounds, I believe, the whole vehicle. And what it is it carries a swarm of robots underneath it. The machine spans 4 strawberry beds, and there's a set of robots on each bed. There's 2 robots on each line of plants. There's 2 lines of plants on a bed. So you have 4 robots on each of the 4 beds. So there's a total of 16 robots underneath the machine. And using vision technology, they're searching for the right berries. And they go in a circular motion around the plant, the robot. It grabs the leaves and holds them upright so it can expose the ripe berries that are laying on the plastic. And then there's a picking wheel. And this is one of the unique things about our technology that our first patent actually was on. is this picking wheel. I call it the Pitzer wheel. I named it after the gentleman that invented it, Bob Pitzer, who is the co-founder of this company, Harvest Crew Robotics. It's really ingenious because what it does is it utilizes conservation of motion. So rather than having a multi-axis robotic arm that picks a berry and then it has to move it someplace, this picking wheel is able to pick very rapidly, because each time it picks a berry, the claw picks it, the wheel spins, and then another claw is there ready to pick the next berry. So as the wheel can go around the plant in about 8 seconds currently, we can pick an entire plant if it has maybe 3 or 4 berries on it. So there's not much time wasted as it spins around the plant.
It wasn't always so in the level of development. It's a fairly sophisticated and efficient machine at this point, but I'm just wondering that there's a lot of parallels between this and SpaceX. I'm just wondering, how many rockets did Elon Musk blow up before they actually had a successful launch, compared to the number of iterations of this robotic harvester?
I like to use that analogy a lot, actually. I've used it in presentations. I mean, people look at the SpaceX Falcon Heavy rockets coming down and synchronously landing on a pad after they've launched the rocket into space, the capsule into space. And, you see that image, but people don't remember all the images of the rockets blowing up in midair and having all these terrible disasters and crashing into the ocean and everything else. So, yeah, this is an iterative process, and I've tried to tell people that you never know, when you go out to the field with the harvester in the first several years, what was going to happen. We didn't blow any up, fortunately, but you never know if it's going to work or not that day. We've fortunately gotten quite a bit past that now. We've got a prototype this year that we believe is going to be the precursor of the commercial machines that are going to be beginning to be built next year. And we're really excited for this coming season here in Florida, because I believe this is going to be the start of what is going to be this robotic revolution.
Plantopia is brought to you by the American Phytopathological Society, or APS, to honor the United Nations celebration of 2020 as the International Year of Plant Health. Healthy plants can help us solve world hunger, stabilize the world's climate, protect our forests, and add beauty to our lives. Now, back to the show. The general public might be accustomed to seeing industrial robots, perhaps from video footage from an assembly plant for automobiles, and you're thinking about that as the state of the art. It would seem that an industrial robot that's welding together a car body has a pretty simple job compared to picking a strawberry by the ton. Exactly.
Yeah, the machines that we're using, and there's got to be some level of intelligence. I mean, they've got to be able to identify the plant and see the berries and tell when a berry is ripe or not ripe, because a strawberry, you have to pick it at that right stage of ripeness, because a strawberry, some people don't know this, it doesn't ripen after it's been picked. So you can't pick it with too much green on it. You want to be able to sort out any overripes. But like I said earlier in the show, you've got to go through there every 3 or 4 days just to pluck off the berries that are at just the right stage. So.
And then there's the whole carriage part of it. The strawberry doesn't come to the robot. The robot has to go to the strawberry in a field.
Yes, and navigate and be able to identify. And so we're recording the GPS location in the field of every plant. So we're re-accessing that center of that plant where that hole was punched, that the berry was planted in, so that it gets recorded when we first lay the field out and punch the holes that we put the plants in. And that's it. And and here in Florida, every October is our month for planting. So all those plants go in the ground, but we're recording millions of plant locations. And so when the machine comes back through, it's able to, very, very precise accuracy, access that exact center of the plant again. So that's a kind of a cool feature of the machine.
And this isn't just a challenge for berry crops. This cuts across a broad range of specialty crops that are presently all harvested by humans.
Yes, it's a big market for agricultural robotics. It's tens of billions of dollars that are being spent on harvesting crops. Strawberries alone there's currently about I think it's $1.5 billion a year being spent on labor to harvest strawberries. So it's that number is continuing to rise too. So it's an opportunity. It's interesting how the stars have kind of aligned. We've got this tremendous need right now to figure out ways to harvest berries or any crop for that matter that requires a lot of labor. So the need is there, but the technology has advanced and has come to the point now where we can do things that we couldn't have done even 10 years ago, or even in 2013 when we started and we're beginning to imagine how we were going to do this. The technology wasn't there yet. The processing speeds. We're taking hundreds of pictures a second of strawberry plants, and the technology to process those images wasn't there when we started doing this even. So it's pretty amazing how quickly things have advanced. But so that the need is there, and then the technology is coming together at the right time that this is being made possible.
Lance, you work on a project called Vitis Gen 2. What's Vitis Gen 2 about?
Vitis Gen 2 is a grapevine breeding program working with public and private breeders across the U.S. and around the world to help develop new varieties of grapes, whether it's table grapes, raisins, wine grapes, juice grapes, that have disease resistance, and specifically resistance to powdery mildew, which is a problem everywhere grapes are grown.
It's my favorite disease, certainly, and it's one of the most destructive diseases of grapevines worldwide. The breeding of resistant cultivars, I've heard it said that it's a numbers game. How does that relate to the work you're doing?
There's been very little grape genetic improvement since the grapevine was domesticated thousands of years ago. Almost all the varieties, 6,000 Vitis vinifera varieties, are highly susceptible to powdery mildew and require grape growers to spray them about 10 to 15 times a year, regardless where in the world they are. So recently, grape breeders around the world have started looking to wild grapes with naturally evolved disease resistance to start to introgress those genes into cultivated grapevine to combine the strong resistance, natural resistance to powdery mildew, along with excellent fruit quality that we know in the varieties we know and love.
So, in a quest for new sources of resistance, when Vitis Gen started, I guess about 8 years ago, we were searching for signs of resistance in collections provided by grapevine breeders all over North America, but it was done manually. Can you describe what that process was?
Yeah, so we would start with the germplasm repositories, the wild natural collections that the USDA maintains and distributes worldwide. And we surveyed those for disease resistance genes and worked with breeders to start to cross-hybridize those with their existing breeding programs and develop what we call mapping families. So these are families that might have 100 individuals in them, 300 individuals. They're all full siblings, right? So they're related to each other. They have the same parents, the same mom and the same dad. And they segregate, half of them maybe having disease resistance and half of them being highly susceptible. They also segregate for traits like fruit quality. Some of them taste awfully nasty and are disease resistant. We want the ones that combine disease resistance with delicious flavors and aromas.
So each of these physical characteristics could be called something. A phenotype would be the technical name for it.
That's right. We use this technical jargon. We talk about phenotypes. Phenotypes are the traits of the vine.
And the process of searching for new and valuable phenotypes is phenotyping.
That's right.
So now we've brought everyone up to speed on the terms we're going to be throwing around. Let's get started on describing the work.
Yeah, so we have this biparental family. Let's say it's 300 individual vines growing in a vineyard, and we need to know which of these vines is resistant and which is susceptible. The traditional way that a grape breeder would do this is just to allow Mother Nature to do its work. Allow the natural inoculum, which is not evenly distributed in a vineyard, to just rain down on the vines. And then at the end of the season, we take ratings of how much disease we see on each of the vines, and that represents the vine's phenotype. The challenge with that method is that Mother Nature is not uniform. It's not a controlled experiment. And we found that year to year, we had highly variable and often unreproducible results. And we had a really hard time identifying the DNA markers that were associated with the disease resistance observed after natural infections.
So the first iteration of the VitusGen project then sought to take some of that variance out of the evaluation process by, in a controlled fashion, inoculating the candidate varieties and examining their response to the powdery mildew pathogen.
That's right. So we would, in the very beginning, receive leaves from a given grape breeder, maybe from all 300 vines in their mapping family. And we would process those by hand. And we came up with a method where we could use 1 centimeter leaf discs as a sample for that grapevine. And we replicate those leaf discs. So maybe each of the 300 vines has 8 replicate leaf discs. And we're able to put about 300 leaf discs on a single tray. This is like a baking sheet with agar in the bottom to keep the leaf discs alive. We're able to inoculate it with 1 controlled genotype of the pathogen and provide an almost completely uniform environment where we're evaluating each of the siblings to compare apples to apples how resistant or susceptible they are.
Initially, though, there was a bit of a hitch in the process in that it was manual and it involved a human observer. So what was that process like?
So setting up the experiment was one thing. It would take a day to create the 2,400 leaf discs we needed for an experiment. And maybe it would be 7 people sitting around a table punching leaf discs and arraying them on trays. But the real challenge was in the manual observation of the phenotypes of how much powdery mildew was growing on each of the samples. So it involved clearing the leaf discs from the chlorophyll so that they appear white, and then staining the fungus so it stood up off the background. You could clearly see this black stained fungus on a white background. And then a human could sit at a microscope and count how many hyphae there were. But each sample took about 5 minutes to look at. And so if you have 2,400 samples, it takes quite a bit of working hours, laborious time at a microscope to read out the phenotypes of a biparental family.
So how many samples could one person examine in a day?
it would be good to examine 30 samples in a day, something like that. We calculated that a typical experiment would take about 1 month or more for a technician to record the data for that one experiment. And we got really outstanding data quality from it, but it took a lot of time and was very low throughput.
Right. And as we said, breeding is a numbers game. So if you want to up the numbers and discover new sources of resistance, that had to change. And so enter the robots.
Yeah, once we figured out the methodology for doing this manually, we realized that with modern automation and neural network analyses, we could really turn up the dial in throughput by automatically collecting images for each of these samples and then feeding those images into a convolutional neural network to quantify how much powdery mildew there is on each leaf disc.
So what were the convergent technologies that came together at this moment that allowed you to go from, say, 30 samples a day to thousands of samples a day?
Yeah, the convergent technologies were really the automation, the moving from one sample to the other, the optics of being able to basically take a microscope image, and the sensor technology of being able to capture all that information in very high resolution. So with this 1 centimeter leaf disc, we basically, each pixel represents 1 micrometer, which is, the size of a bacterium. It's very, very microscopic, tiny stuff we're looking at. And then, of course, the artificial intelligence technologies are amazing. And we found actually that the computer vision approaches that we're doing, these neural networks, they're more accurate than the human sitting at a microscope in quantifying powdery mildew and predicting what genes might be underlying the resistance.
So the device to do this is called the Blackbird, and it's got an interesting history. It's named after a Cold War spy plane.
Yeah, well, it's very much like a surveillance plane. It's just on a completely different scale. We have this camera hovering up over top of this leaf disc sample with these tiny fungal threads that are growing. And it's basically a surveillance of what's happening with those hyphal threads. A beautiful thing that this imaging platform allows us to do is, whereas with technicians, we needed to kill the powdery mildew samples and just take a snapshot of what they're doing, with this imaging robot, we can do live imaging and watch how the fungus grows over time, repeatedly coming back to the same samples and seeing how they've changed.
And so the promise of this high throughput system was the discovery of new sources of resistance. Has it done that?
Absolutely. So, as you said, it's a numbers game, right? So before, we were able to, in a single year, conduct anywhere between 6 and 8 experiments. Last year, we were able, with these imaging robots, to conduct over 30 experiments, and we identified 12 new resistance genes thanks to this throughput. So just to give a little context, in all the previous work of the past 20 years, working on grape powdery mildew resistance around the world, in 1 year, we doubled the number of resistance genes that are known.
You've taken an off-the-shelf, high-grade commercial camera, something that is accessible to a consumer, equipped it with an appropriate lens, and it's been combined with the stage of a 3D printer for movement and precision movement. And then the images that are captured are run through a convolutional neural network for analysis. So each of these steps is something that we would not have had access to, say, 5 to 10 years ago. It's all a result of very recent developments.
and those developments are continuing to push the forefronts of what we can envision. I'm very excited this year. We're going to be adding on thermal imaging and see what information we can gain about disease progress in relation to the thermal signature that the fungus and host release. We'll be adding hyperspectral imaging so that we can look at the biochemistry of the interaction during disease progression. There's just so many things, things that we can pursue.
So currently we're capturing color images at a very high resolution that allow us to see something formerly only observable at very high magnification under a microscope. It's being captured by a consumer camera.
Right. These are very high resolution, 50 megapixel consumer cameras, and those sensors are only going to continue to improve. So the resolution of what we might see 2, 4, 5 years ago or from now is only going to get better. So not only are we thinking about how far we've come in the past couple years and how quickly we've gone from low throughput manual data collection and analysis to what's currently blowing our minds with the high throughput and how quickly we can go from a hypothesis to a gene. And the future is really exciting.
What's the next bottleneck?
Well, now what we've been doing, of course, is just looking at 1 centimeter leaf discs in a highly controlled environment. The next exciting thing is taking the technology out to the vineyard and doing field work to look at natural disease incidents, early detection of pathogens, and quantification of those pathogens on leaves that may be different sizes, different textures, at different angles to incident light. Much more complicated problems exist for us in the vineyard.
For more information about the International Year of Plant Health, visit us at plantopiapodcast.org. Thanks for listening. Our show is produced by John Bryce. Thanks also to Mark Gleason, Jim Bradeen, Laura Isles, and Roshni Karati. I'm your host, David Godori, and you've been listening to Plantopia.